The Cell Cycle by Adira Chakhanal. Eukaryotic cells go through a set of stages that determines whether the cell turns out normal or not. This process is called the cell cycle. The cell cycle has some very important phases. Phase 1 is called interphase. Interphase is the longest phase of the cell cycle. It has 3 subphases: G1, S phase, and G2. The next phase is called M phase, represented here by 5. M phase can either be mitosis or meiosis, depending on the type of reproduction. 6 shows cytokinesis, which is the division of cytoplasm. To make sure the cells divide correctly, they are regulated with checkpoints that come up in the cell cycle. In this diagram, the checkpoints are represented by the red. These checkpoints give stop and go signals. The signals indicate whether crucial processes have occurred by that point in time. If not occurred, then the cell cycle will stop completely. There are 3 major checkpoints that are found in the cell cycle: G1, G2, and M. G1. Cells pass this checkpoint when they are stimulated by appropriate external growth factors. G1 checkpoints are controlled by cell size, growth factors, and environment. G2. This is the checkpoint to make sure no damage has been done after DNA replication. DNA replication occurs in the S phase. If damage has been done after DNA replication, then it prevents the cell from continuing on to the next phase, which is the M phase. It prohibits it. M. This checkpoint assures that the microtubules are attached properly to the kinetochores. Microtubules are just another word for spindle fibers, or they're what make them up. Kinetochores are right here in this diagram. They are made up of proteins and are the anchor sites on chromosomes where the microtubules attach. So here is the kinetochores, and the spindle fibers would attach on both sides to make sure that the chromosome moves where it's supposed to during the phases of mitosis or meiosis. If the spindles are not properly attached, then they do not continue through mitosis or meiosis. So, all in all, G1 regulates cell size, nutrients, growth factors, and DNA damage. G2 regulates cell size and DNA replication. M makes sure that the spindle fibers are attached properly to the chromosomes. There are also internal checkpoints. Complexes made from specific proteins tightly control cell division. The complexes contain enzymes called cyclin-dependent kinases, otherwise known as CDK. In this diagram, CDK is shown as this yellow block. The function of CDK is to turn on or off the various processes that take place in cell division. So, they basically control the cell. Most kinases stay inactive until it attaches to the cyclin. In this diagram, this purple block indicates cyclin. Cyclin-dependent kinases, hence where the name came from. MPF, or mitosis promoting factor, contains cyclin A or B. It is shown here on this diagram. It is the combination of CDK and cyclin. When the level of MPF exceeds the strength needed to trigger mitosis, it is turned on. This indicates that G2 has ended and mitosis has started. There are also external checkpoints. There is a protein released by certain cells that stimulate other cells to divide, called growth factor. Growth factor plays a role in external signals because most animal cells don't divide unless the growth medium includes specific growth factors. An example of this would be a platelet-derived growth factor, or PDGF. When there are disruptions in the cell cycle, cancer cells are formed. The faulty checkpoints make the cell unable to check stages for disorder. They form from the mutation of genes in the cell cycle. Cancer cells don't exhibit density-dependent inhibition. When this occurs, the cells stop dividing altogether. They keep growing and dividing despite the crowding or detachment. They are capable of doing this because they either do not need growth factors or they make their own. Cancer cells have no control system, so they can stop growing whenever they want. They can also start growing whenever they want at whatever speed they want. In this diagram, it shows cells forming a tumor, which is cancer. You can see how disorderly and crowded it looks here, while in the normal cells they are orderly and next to each other with no collision. You can also see in this diagram how they are formed, how this is in orderly pairs and patterns, but this is all crumbled together. This concludes how the cell cycle is controlled by internal and external mechanisms, and how the loss of this control can lead to cancer.